Okay, thanks very much. Again, I'm Justin Vaughn, and I'm from the University of Georgia. I'm going to be talking about, the biology of naturally occurring insertions, and of course this is to contrast to events we see with regard to transgenic plants or genetically engineered plants. Before I get started, I'd really like to thank the organizers of this conference, and also to Nancy and Brianna for organizing the organizers. And this is my first time in Canada, actually, and I've gotten a chance to walk around Ottawa quite a bit, and it's a beautiful city, a great introduction to the country, particularly all this kind of white substance that the natives call snow. We don't get much of that in Georgia, particularly South Georgia here, and it turns out that a lot of our plants tend to like those hot, humid conditions. So we have peanuts and pecans and cotton that grow down there quite happily, but it also turns out that a lot of pests and mildews also tend to like those hot, humid conditions. Particularly with regard to our poor state fruit here, we're called the peach state. It's plagued by numerous pests and mildews and those types of things that I don't know if anyone's priced an organic peach lately, but I think they run at about $5 a pop or something like that. So it's if you can find one. So we would, given the fact that fruit tree breeding is generally something that occurs over the course of human generations, we would really like techniques that advance tree breeding in particular. So before we begin to adopt those techniques wholesale, of course, we need to identify both the intended and unintended effects of genetic engineering, which is, of course, what this meeting is all about. For the purpose of this meeting, I've defined three sort of general categories as sources of unintended effects, and this is echoing things that people have already said, but just to reiterate, because this is the first talk of the morning, we have this possibility for an unintended function of the actual transgene sequence, right? So our transgene sequence creates a protein that has the intended effect, which is pest resistance or what have you, but it also may have an unintended effect, such as upregulating pathway X, as exhibited by this red gene here. Oops, sorry. There's also the possibility for structural rearrangement at the site of transgene insertion. And of course, this also includes situations where the transgene jumps into a pre-existing gene of known function or unknown function, hypothetical gene, and disrupts the physiology of the plant. It also includes situations where we have DNA from somewhere else in the genome, here shown as DNA from a different chromosome, gets moved into the site of transgenesis. A third possibility here is that we get a clean event, but the process that went into creating the event encourages the movement of genes from their original locus to other regions of the genome. I'm going to be mainly emphasizing this type of unintended effect and the mechanisms that go into this. And I'll kind of touch on these, and I'm going to completely ignore these because I'm sure there are a lot of talks related to that. So what is the real molecular nature of these effects? As I've said already, if we have the transgene that jumps into a gene, we get a knockout, and tDNA has classically been used as a mutagen for Arabidopsis to find and characterize the function of genes in that model system. Chryson et al. Defined this knockology here because we have these alternative events such as reduced expression, increased expression, and these other situations here. Of course, all of these can basically be tracked down in terms of where the position of the tDNA insertion occurs. So if it's proximal to a gene, you can pre-screen these to avoid these events to some extent. I know there's been talk that, well, even if it isn't a gene in the plant, the corn is still corn and what have you, then why worry about it? But I think still the consensus is that you want to try and avoid putting your transgene near a gene for genetic regulation reasons, among other things. Now, a little bit more complicated is the fact occurs when we have the transgene associated with a rearrangement, because even if this occurs at a site in the genome that doesn't involve a known gene, we can still pull in genetic DNA or gene encoding DNA into that site. So that's kind of what I'm going to be talking about a little bit. And you also have the possibility of creating chromosomal translocation, so major structural rearrangements. And Katie Clark and Patrick Chryson looked at this and 64 different lines and actually found 12 translocation events. And they did this by kind of classic genetic mapping techniques, showing that the recombination frequencies between markers differed from what they expected by chance, and that these were indicative of translocation. Now, it turns out that almost all of these events were caught, as they would be, I think, in a normal breeding cycle, as pollen phenotypes. So because of the pairing between homologous chromosomes that has to occur at meiosis, you get this abnormal pollen phenotype. There was only one of those that escaped this phenotyping. And I'd like to emphasize too that it's not clear that the tDNA insertion was actually the cause of these translocation events, but it's just that 64 lines, tDNA lines, had these 12 translocations. But even when we have a single copy tDNA insertion and no translocation, we're still getting rearrangement at the site of insertion. And so this is kind of a classic paper here where Forsbach et al. Went in and sequenced the junction sites of single tDNA insertion, so a fairly well-behaved situation. What they find, these two top categories here, are situations where you basically get a near perfect insertion without any additional DNA moved into the site. And for the left border, that accounts for about 50% of the time, and for the right border, that accounts for about 30%. About another 50% actually end up bringing in at least some what they call filler DNA here. So the majority are very short filler sequences, and then we have some that are fairly substantial in length, at least could encode a motif or something to that effect. And then we have a couple involving the right border, which are actually quite large. And so these are commonly cited as concerns for bringing in basically uncharacterized DNA into the site of transgenesis. And of course with bialistic transformation, because, for a number of reasons, because the DNA is not getting guided to the nucleus, and it's also being moved into the nucleus in much higher concentrations, we typically get more complicated events, although I think it's still an important technique because of the peaches and things like that I talked at the very beginning. But the whole point of this talk is how do these compare with structural rearrangement in plants and crops? And of course, I think Wayne hammered this home pretty well with some really nice examples yesterday. I'm going to kind of get into a little bit more quantitative detail and some of the mechanisms by which this occur, and then contrast those with tDNA insertions. So a couple years ago, Michael Lynch's group, they took 5 plants and they advanced them 30 generations by single seed descent, and they identified that within this population, 150 plants, they had 4 large deletions of substantial size. So some of these are 5,000 base pairs, 600 base pairs, and so this gives you a sense of just with a selfing plant, a well-characterized genotype, and greenhouse conditions, you still get substantial structural variation. And this actually ignores, because of the for technical reasons, this actually probably ignores the larger insertions that you might find because they were using very short sequencing reads, and I'll talk about that a little bit more in a second. So this kind of goes on to show that you also get these insertion events. What this group here done, pervasive gene content in maize, is that they took the genomic DNA and basically hybridized that on a gene chip. And so what we're only looking at here is genic space, and they're looking at up copy number variation shown as these blue aberrant points, and down copy number variation shown as these red points here. And you see that there is clearly in maize dramatic differences in entire gene content. And this is just a blow up of one of those chromosomes. So as I said, this is primarily genic space right here, and so we'd also like to look at non-genic space, and we'd also like to look at nucleotide level resolution to really tell us what the mechanisms are that are going on in terms of these rearrangement events. And so the way we can do that now is through next generation sequencing data. We can essentially resequence the whole genome, excluding centromeric regions and some other elements. So the way we do that is we have a read here shown as green. So this is a sequencing read of 100 base pairs or a little bit longer now. And another one here that's a pair, and there's this spacer in between that's not, it's defined maybe within 20 base pairs in terms of plus or minus 20 base pairs. We think it's 300 plus or minus 20 base pairs. So we have a rough idea of the distance between these two things. When there's no structural variation, we can map those. When there's a deletion, we map those, and it throws up a red flag because they map in a way that's different than our reference. We can also, and I'll talk a little bit about this technique later, you can map a region of the read to a known locus in the genome, and then the rest of that read may map to a known mobile element or a tDNA insertion, for example. You can also look at tandem duplications. Complementary data is that we see a difference in read depth over that region, so these sort of complement one another, and also in terms of deletions, where we see missing reads. And of course, a read can be split across a deletion relative to the reference. Finally, one of the most useful things for us at least is that you can assemble these reads de novo and then map those to positions in the genome. And this gives you basically nucleotide level resolution of novel insertions. And we don't have to know the insertion beforehand. We can actually find out what the new sequence is. So what we did is we looked at these using this technique in cultivated rice population. So this is just a phylogenetic tree here. Indicas are shown in blue, japonicas are shown in red. So we have about 50 varieties of cultivated plant here. And we inferred whether these were insertions or deletions based on looking at an outgroup, so we could get really defined characteristics. And so what we find here, we actually ended up looking at 4 chromosomes. These represent 131 megabases, so that's about a sixth of the Arabidopsis genome. Across that entire population, or of those 4 chromosomes, we find 65,000 structural variations. So these can be, and these are between 10 and 1,000, even maybe 10,000 base pairs long. So this is actually ignoring really short indels, which would, of course, increase those numbers dramatically. The deletions and the insertions are actually fairly balanced. So as I said earlier, a lot of times older techniques would miss these insertion events. We actually see, actually a comparable number of insertions. And I'd like to emphasize here that these are actually these structural variations are events that are shared between multiple cultivated lines. So individual lines will have their own events too, and that probably accounts for another large fraction of these structural variations. So again, this number is going to increase as our resolution increases. So what do these events look like? So if you imagine here, this is an insertion site, okay, and this is about 300 base pairs upstream and 300 base pairs downstream from that insertion. And what this is showing is the DNA that was copy and pasted basically into this insertion site. So what this is showing is basically that there are a large fraction of events where the insertion comes from very nearby. For example, here we have 30 base pairs that is copy and pasted from 170 base pairs away into this site, and 100% of that insertion is explained by that event. There are other situations where only half of the insertion comes from nearby and the other half comes from somewhere completely different. Or situations, and this is interesting to see, where we get an insertion partly from upstream and then partly from downstream. So fairly complicated events can be uncovered in this analysis. So we kind of summarize all that information, and you can ignore these parenthetical statements here. I'll try and explain those. But these warm-colored portions of the graphs here are those local duplications that I just showed you. So this y-axis here represents all the different length categories. So here are 10 to 19 base pairs, and this represents just the total number that we observed. So these black and gray bars actually represent perfect tandem duplication events. So that at the site of duplication, that perfectly adjacent neighboring DNA was copy and pasted into that site. And then we also have partially tandem. So this includes the DNA right next to the site, but also some type of filler DNA. And then, of course, the local that I described. And then finally, these white bars represent DNA that we couldn't track down. It may have come from somewhere else in the genome or just randomly ordered DNA. So these naturally occurring structural variations do have an effect too. So this is a nice genome-wide association study done by Ben Hahn's group in China, where they actually were able to pinpoint causal variants that explain phenotypic variation in the rice population using these powerful statistical techniques. And so they associated this causal genetic variant with some phenotypic variation. And here you see this is an example of where they had a 23 base pair duplication could explain a portion of this phenotypic variance. And it turns out that these are actually quite common. Here's just a 10 base pair deletion. Here's a 14 base pair deletion. And then, of course, you also find SNPs that are associated with these phenotypes. I think it's interesting that when you normalize for the number of these polymorphisms, that you find many more structural variations in relative to SNPs. So when you correct for the number that appear in a population, they seem to have a much more prominent role in generating phenotypic variants. I think also some of Wayne's slides support this idea, since we've discovered that so many of the genes underpinning traits that we've observed or created are related to structural variation. To kind of hammer the point home, what we've actually we've looked at a sort of genome-wide analysis. A couple of years ago, my former boss, Jeff Bennetson, looked at a similar type profile as this in 1 megabase of DNA, so 1/600th of the genome of rice. And as we discovered here, like the tandem duplications that I kind of talked about, you remember how much of the graph that those took up in my slide. But he also discovered a large fraction of insertion LTR retrotransposons. So these are kind of groups that are flying under the radar of our analysis, and they're still very difficult to detect the insertion of these in a genome using deep sequencing, next generation sequencing technologies. So, and actually, so just as large a fraction of the genome is accounted for by these events, And actually, in terms of total size, the vast majority is accounted for by these events. So we're still just on the tip of the iceberg in terms of identifying naturally occurring structural variations. And these are both, these numbers all come from a comparison of indica to japonica for a region of the genome that aligns across 1 megabase. So I think this is particularly in terms of the transposon movement story. Even when you look at a single rice genotype that's been taken, induced to culture, brought back to a plant, and then advanced for 2 generations, and you can now map those transposable elements via the technique that I talked about earlier, where you map a read to the genome and then you map your paired read to a known transposable element. This group was able to uncover numerous examples of transposable element that had occurred just through that generation cycle. So it appears to be a major source of naturally occurring structural variation, even in a single genotype. And so this also has an impact on secondary impact, these transposable elements when they move in the genome. And this is sort of postulated by Thomas Wicker, where they looked at where transposons where they actually looked at where genes had moved in a comparison between rice and sorghum. And they found it was very common that these genes had moved proximal to transposable elements. And so their model, and there are other evidence associated with this, but their model was basically that the transposable element movement disrupts the local DNA and makes it more susceptible to double-strand breaks. Those breaks get repaired by sampling from somewhere else in the genome and pulling these genes into the position to which they're associated. So again, not only do you have the transposable element activity, you also have the potential for creating double-strand breaks and bringing endogenous DNA into the site of insertion. So we wanted to test this in the absence of a transposable element. What does it look like when we just create a double-strand break in the genome? And so what we did is we took a maize plant and rice lines as well, and we had an enzyme that cuts at a very specific site. So this is a 17 base pair site, and this enzyme will only cut at that site. So what we do is we cross these plants and we examine the results of that cross with PCR across this cut site, and then when we see what these events look like. And so it turns out that actually a large fraction of these are deletions, and that we get about 20% are insertions coupled with deletions. So typically you're going to be looking at small events being deletions. There are actually some larger events that we missed, and so we went back and looked at some of the larger events and tried to characterize those a little bit more. And so what you see here, and I know this is a little bit of a complicated slide, this is from Sujin Kwan's unpublished work from our lab. And so you see what's represented, for example, in this case, is that 8 base pairs from the 38 base pairs away was moved into this site, along with 34 base pairs from 335 base pairs away moved into this site. And so if you recall from looking at the natural structural variation, this is almost exactly what we see in that case. We see these complex events. So we think that the double-strand break repair and these two experimental and comparative genomics approaches are really telling us the same story about how these sites get repaired. And also a group a couple years ago looked at, actually more than a decade ago, looked at a similar, using a similar system, looked at really large events and found that that these, typically are sampling. Basically, this is what you'd expect randomly if you looked at the tobacco genome, what you would expect to randomly pull out from that genome if you're going out into the genome and sampling DNA into your cut site. And it also, to me, this is important that the cut site in and of itself doesn't activate a transposable element. You don't see any intact transposable elements. Here you do see one retrotransposon sequence, but this is just a fragment that appears to have been copied from somewhere else in the genome. So these do occur when you create double-strand breaks in an experimental system. So this really leads us to a couple models of double-strand break repair, and also to some frequencies for how common these occur and how they're repaired, the frequency of how they're repaired. So when you get a simple break, actually the most common thing that happens in a eukaryotic genome is that these ends just get blunt ligated together. And so that's basically what's shown here using these known proteins that bind to the ends of those break sites. If for whatever reason this break doesn't get immediately repaired, you also have this common pathway where you use microhomology between the two ends of the break site to bind to one another, and you get a deletion of that intermediate DNA. And less common, but clearly, as I've shown, this occurs and possibly more frequently than we think. This is 5% is probably a more accurate estimate, is that the break site goes into somewhere else in the genome, copies a portion of that, and then comes back to the break site here and binds in, and you get a gene, basically a gene movement, or at least a sequence movement event. And it's also possible that this sequence can copy from its neighbor here, and that seems to be actually what we're finding most often occurring. And so I think this is pertinent to tDNA-induced rearrangements, because we actually see a very similar process. So here we've seen structural rearrangements when we induce double-strand breaks, and also when we have tDNA that is, basically, if you'll recall this slide where the junction region between the genomic DNA and the border of the tDNA sequence was sequenced, and the number and the spectrum of events that we observed there match quite closely with what we've observed in natural populations. And so this is kind of reiterating those points. Structural variations are not unique to transgenic plants. I think we've all agreed on that. The double-strand breaks and transposon activity result in structural variations in essentially every plant, given the frequency of these events that we've observed. Every one of those rice varieties have major structural variations, and that the tDNA-induced rearrangements have a pattern of mutation that is really similar to what we observe in structural rearrangements in the natural populations at a mechanistic level. So hopefully these mechanisms will help you as regulators, also give you some idea of what potentially might be coming down the pike in terms of new generation of transgenics, where we use homing endonucleases to actually create these double-strand breaks, which we can create gene disruption events, point mutations, or even gene deletions or gene additions. And these, again, are acting, since we observe double-strand breaks throughout the genome, we anticipate that the results of those structural rearrangements will be the same for potential new generation of transgenic plants. So with that, I think I have about the right amount of time. I'd like to thank Jeff Bennetson, who is actually my just former boss. I've actually moved to the soybean molecular breeding program there. It's still at UGA, but sort of a new home. Sujin Kwan did a lot of the experimental work, and also Feng Liu, who's not pictured here, did a lot of that unpublished SCE1 work. So with that, I think I will leave the floor to Thomas. Thanks.